Continue on with our PowerPoints for this week. We have Chapter 6, which is going to discuss conception and prenatal development. Our first slide here, we can see the gametogenesis, which is the formation of the female gametes beginning during prenatal life. When the oocyte matures, two meiotic divisions reduce the chromosome pair from 46 pairs to 23 unpaired chromosomes. After that, we can see the gametogenesis and the spermatogenesis, which is the formation of sperm or the male gametes in the testes. Each spermatome contains 46 paired chromosomes. The first meiotic division forms two secondary spermatocytes and reduces the number 23 unpaired chromosome in each gamete. So each individual gamete has 23 unpaired chromosomes. The gamete from a male determines the gender of the new baby status post conception. On our last slide here, in slide 4, we can see a closer visualization of the sperm gamete itself, which has the head that contains the nucleus with the 23 chromosomes, the midsection, the tail, which help motility and movement. Conception is the preparation for conception in the female is the release of the ovum and the ovum transport, or the transport of the egg itself. Conception also includes preparation in males, ejaculation, again, transport of the sperm. We saw that in that previous slide where we got a better visualization of that tail for movement. Fertilization, both entry and fusion. And if you click on the conception part, you get a video, which I highly recommend watching, of conception formation itself. It's nice to be able to pull in what is actually happening during the time of conception in a more visual manner. We can see a picture here on slide 6 of fertilization, both the entry point, the fusion, and the fertilization complete in section C there. That's the complete new embryo we can see. Transportation, remember reviewing that female preparation for conception there. So fertilization can occur, status post-transport here. We can see that zygote moving further down the fallopian tubes until it implants into the uterus. And we can see that implantation in the larger photo there, more up close one of the zygote itself. Pre-embryonic period is from fertilization to 2 weeks after. During this time, cells are rapidly dividing. We have the entry of the zygote itself into the uterus, which we saw a visualization of in the previous slide. We have implantation into the uterus, location of implantation, and the mechanism itself. And here is a nice visualization of that embryonic period. Again, that differentiation of cells that we can see in about weeks 2 to 3. And then weeks 2 through 8, we have more of the development of from a zygote to more of a fetus that we can see. We start to see the development of the eyes, the ears, the umbilical cord, remembering that the umbilical cord is going to be adjacent to where the placenta is going to implant as well into the uterus. As the weeks of pregnancy continue to evolve, the fetus itself continues to develop. We can see a more kind of better developed eye in weeks 6 and 8 versus weeks 4. Fetal development period, again, if you click on that link that says fetal development, blue highlighted underneath there, you get a nice video of the development of the fetus from early on in pregnancy, status post-conception, through a term pregnancy. Term begins at 37 weeks. A full gestation is 40 weeks. Remember that pregnancy is measured in weeks, not months. Slide 11, we can talk about the auxiliary structures, including the placenta. And that is going to be the communication between the baby and the mother. Communication in terms of circulation, bringing nutrients, taking away waste products. So it plays a really important role in fetal development. Maternal component of the placenta, remember that it is going to be kind of like almost stuck, if you will, or if not more embedded into the uterus on one side of it. The other side would be the fetal side the placenta will develop with the fetus, so it's going to grow with the fetus in order to support circulatory needs. Fetal component, again, talking about that fetal side and reviewing those metabolic functions, again, bringing in nutrients and taking away waste products. The placenta is also responsible for gas transfer, waste removal, antibody transfer, transfer of maternal hormones from the mother to the baby, as well as overriding the endocrine functions for the fetus. On slide 13, we can see a lateral view of the placenta here. We can see towards the bottom of the photo, if you look at the fetal circulation area, that would be the umbilical cord, which is going to be the main communication port between the placenta itself and the baby. And again, remembering the maternal side and the fetal side. They both will allow for circulation. One talks about where the placenta is implanted versus where the fetal side meets. And the other side over here on slide 14, we can see a better visualization of the placenta itself from the amnion side. We can see implantation of a healthy umbilical cord towards the center of the placenta itself. On the right-hand side, we can see more marginal placement of an umbilical cord that is more of a placement of the cord towards the border of the placenta versus in the center. Towards the bottom left side of the slide, we're looking at a photo of a placenta with a small accessory lobe, and we can see that kind of adjacent bubble next to the semicircle, if you will, of the placenta itself. Most commonly, placentas are sent to pathologists status post-delivery so that they can be analyzed by pathologists to make sure they're intact, that there's no additional lobes that we should be aware of, because that can cause bleeding in moms postpartum. So it's important to know what a placenta looks like as a baseline in order to be able to assess if there are any changes that someone needs to be aware of, providers need to be aware of. The bottom right-hand corner again of this slide, we can see a filamentous insertion of the umbilical cord, and that's one where the umbilical cord branches out in its own particular membrane. This can cause issues in circulation from both the baby and the mom. As we can see, it's not a solid implantation like we saw in our first photo of the placenta. Let's review a little bit more of the auxiliary structures. Fetal membrane and amniotic fluid. Again, both things that are going to be present in a gestated uterus. Fetal membrane, that's looking at the placenta. Again, the amnion is going to be the inner membrane. The chorion is the outer membrane. Those are the sides of the placenta there as well. Amniotic fluid cushions against impact during into the maternal abdomen. So think about it as an extra barrier from baby to the outside world. It helps the environment in the uterus to maintain a stable temperature. It allows for symmetric development. It prevents the membranes of the placenta from adhering to fetal developing parts. As we saw in our earlier slides, we know that zygote is going to hopefully continue to develop into a fetus, and they need to be able to have room in order to continue that development and be able to grow. Amniotic fluid also allows for room and buoyancy for fetal movement. Towards the end of gestation, fetuses practice swallowing and breathing movements with that fluid as well, so it really plays a vital role in fetal development. Slide 16 takes a picture here of fetal circulation. Fetal circulation is going to be different from an adult or a pediatric circulation process, because we know we have that accessory organ of the placenta that is going to be our main communication from mom and the baby. So it's important to understand the differences in fetal circulation than in a pediatric patient or an adult. Fetal circulation includes the umbilical cord. The umbilical cord has typically 3 vessels. A nice acronym to remember those vessels is AVA, artery, vein, artery. So the umbilical cord has 2 arteries, which contains the deoxygenated blood, and 1 vein, which contains the oxygenated blood. Fetal circulatory circuit. The circulatory circuit includes the ductus venosus, the ductus arteriosus, and the foramen ovale. These structures themselves play a role in bypassing the fetal lungs in the fetal circulatory system. And we know that these babies growing in utero and developing in utero aren't breathing in oxygen like you and I are. So they do not require these lungs to have the relationship that they do in a developed human body. Moving forward to slide 17, I would highly recommend clicking on that link there that says fetal circulation. This is a great visualization of the fetal circulatory system. It will bring you to a YouTube site and really highly encourage pulling in some of those videos as well. It really helps to pull in some of the information that you learn both in your reading as well as during lecture. Continuing on with fetal circulation, we understand that the circulatory system of a fetus is affected status post-birth. Those structures that we needed in utero to bypass the lungs are going to be impacted status post-birth as the baby starts to take in oxygen from the room. Changes after birth include fetal circulatory shunts are not needed. Again, reviewing that information I had stated previously. No longer do we need to bypass the lungs, and we also no longer need to circulate blood to the placenta. So that's going to impact the fetal circulatory system after birth as well. Slide 19, we can take a look at multifetal pregnancies here, so that it'd be more than one zygote developing and implanting. And we get a nice little picture there of two zygotes during implantation in our model B there. Monozygotic is a single ovum and a single sperm with later division developing into one fetus. Dizygotic are two ova that are fertilized by different sperms, so they're individual zygotes, and high multifetal gestations there. Her final slide is another question here. How does the placenta allow exchange of oxygen, nutrients, and waste products between the mother and the fetus? So hopefully after the completion of this PowerPoint, as well as your class readings, you'll be able to answer this with success.